Hello students, thanks for watching Edupedia word videos. My name is Niyati Seth, and in this tutorial we will continue to discuss about the other classification of protein where we have left in our other previous section of this chapter. Proteins may be classified as simple proteins and conjugated proteins. Simple protein, it is composed of amino acids that are building blocks of protein. So they are simple amino acid. Examples are histones and albumin. This is the representation of a simple protein. Whereas the conjugate protein, it is formed from the combination of peptide chain and cofactor. Okay. Remember, from a peptide chain combination with the cofactor. Now the simple protein. Simple protein consists of only amino acids or their derivatives. When hydrolyzed by acids, alkalis or enzymes, simple protein yield only amino acid or their derivatives. They include the following groups. One is albumin, globulin, glutenin, prolamines, histones and protamines. Albumin, they are water soluble proteins that are found in all body cells and they are also in the bloodstream. Okay, albumin, they acquire a shape of a sphere. Examples are lactoalbumin that is found in milk and serum albumin that is found in blood. Now come to globulins. Globulins, unlike albumin, they are insoluble in water, but they are soluble in dilute salt solutions of strong acids and bases. For example, lactoglobulin that is found in milk and ologlobulin. Third is glutenins. They are soluble in dilute acids and alkalis. The protein glutenin from wheat is an example. They occur only in plant material. Fourth is prolamines. They are soluble in 70 to 80% alcohol. They include fliadin from wheat and zein from corn. They are found only in plant material. Then histones. Histones are water-soluble proteins in which basis amino acid predominant. They are rich in arginine or lysine. In eukaryotes, the DNA of the chromosome is associated with histone to form nucleoproteins. Okay. Protamines. Protamines, they are also water-soluble proteins. They are basic polypeptides with a very low molecular weight, around 4000 Daltons. They are very rich in amino acid arginine. This is the distinguishing feature of protamine. The polypeptide chain, they consist of 28 amino acid residue which include 19 arginines and 8 to 9 non-basic amino acids. Protamines are found bound to DNA in spermatozoa of some fishes. Now come to another classification, another class of the classification of protein, that is conjugate protein. Conjugated proteins, they are the complex proteins which on hydrolysis yield not only amino acids but also other organic or inorganic components. The amino acid, the non-amino acid portion of a conjugated protein is called as prosthetic group. Unlike simple proteins, conjugated protein are classified on the basis of chemical nature. These are nucleoprotein. Nucleo means nucleic acid and protein means protein. So protein plus nucleic acid. Mucoprotein or glycoprotein means, glyco means carbohydrate and protein is protein. So carbohydrate plus protein. Chromoproteins, where chromo means colored pigment, and lipoproteins where lipo means lipid, so lipid plus protein. Metalloproteins, a metal that binds protein combined with iron, copper or zinc. And then phosphoprotein where the proteins are attached with the phosphoric acid group. So where are proteins present in our body? In blood. The hemoglobin protein carries oxygen in your blood to every part of the body. are present in enzymes in your saliva, stomach, small intestine, and are protein that help you to digest food. Proteins are also present in antibodies. So antibodies are protein that help defend your body against foreign invaders such as bacteria and viruses. Okay. Cellular messengers, in cellular messengers also they are present in receptors. So receptor proteins, they stir the outside of your cell and transmit signals to partner proteins on the inside of the cells. In muscles, they are also present. Muscle proteins such as actin and myosin, they enable all muscular movement from blinking to breathing to roller braiding. They are also present in hair and nails. A protein called alpha keratin forms your hair and fingernails and also is a major component of feathers, wool, claws, scales, horns, and hooves. Okay. They are also present in brains and nerves because there are proteins that are involved in channeling. So ion channel protein, they control the brain signaling by allowing small molecules into and out of the nerve cells. Okay. What are the biological importance of protein? Proteins, they are the structural components of cells. Second, the biochemical catalyst known as enzymes are proteins. The protein known as immunoglobulins, they serve in defense against infections. Many hormones such as insulin and glucagon are also proteins. Protein, they participate in growth and repair mechanism of body tissues. A protein called fibrinogen helps to stop bleeding. Oxygen that is transported to different tissues from blood by hemoglobin, which is a protein attached to the heme part, is a hemoglobin. Okay. Types of protein. There are 8 types of protein. One is enzyme. Example is amylase that helps in the digestion of our food. And amylase is present in our saliva. Second type of a protein is a transport protein. Like a hemoglobin, they help in the transportation of oxygen into other tissues of the body. Myoglobin, that helps in the transportation of oxygen in muscles. Albumin, they help in the transportation of fatty acids. And lipoprotein, they help in the transportation of lipids. The other type of protein is storage protein. Example is ovalbumin, milk, and ferritin. Ovalbumin function is egg white protein. They help in the iron storage in spleen. Spleen is a graveyard of dead RBCs. Contractile protein is myosin and actin that help in the muscle movement. The other category or type of protein is protection, such as antibody. They help fighting infection against antigens, and fibrinogen or thrombin, they help in the clotting of the blood. Hormone protein examples are insulin and growth hormone. Function are carbohydrate metabolism, growth, and regeneration. Structural protein are glycoproteins, collagen, and elastin. They help in making of skin, tendons, bones, cartilage, ligaments, and cell wall. Toxins are example is Clostridium botulinum or ricin or snake venom. Function is botulism, food poisoning, castor bean toxin, snake venom. There are at least 7 functions of protein all in all. Enzyme catalyst, they act as a enzyme catalyst which is specific for only one reaction. Second is defense, such as antibody, protein, other proteins. Okay. The third is transportation. They help in the transportation, such as hemoglobin and myoglobin transference, etc. Support, such as keratin, fibrin, and collagen. They help in motion, such as actin and myosin. They help in the muscular movement. They help in regulation. Some hormones, such as regulatory proteins on DNA cell receptors. The other function is the storage. Calcium and iron attached to the storage proteins. Okay. Now come to concept of metabolism. Metabolism, you can say that metabolic pathway can lead to a more complex structure from a simpler structure. For example, acetic acid becomes cholesterol, or lead to a simpler structure from a complex structure. For example, glucose becomes lactic acid in our skeletal muscle. The former case are called as biosynthetic pathway. They are also known as anabolic pathway, and the latter constitute about degradation, and hence they are known as catabolic pathways. Okay. Metabolism is of two types, linear and circular. Okay. In linear pathway, as you can see that the initial substrate got converted into intermediate 1, and that got converted into intermediate 2, 3, and finally end product. So enzyme is B is specific to the initial substrate. This is changed to intermediate B, and enzyme is specific to intermediate 1 that converts this to product intermediate 2. Okay, and so on. The presence of enzyme, they control the pathway. Okay, what we have learned till now is that living organism, be it simple bacterial cell, protozoan, plant or animal, certain thousands of organic compounds. These compounds or biomolecules are present in certain concentrations. One of the greatest discoveries ever made was the observation that all these biomolecules have a turnover. This means that they are constantly being charged into some other biomolecules and also made from some other biomolecules. This breaking and making is through chemical reactions constantly occurring in living organism. Together, all these chemical reactions are called as metabolism. Okay, and they are of two types, linear and circular. Now come to metabolic basis of linking. Metabolic pathway can lead to a more complex structure from a simpler structure that I have told you just before. For example, acetic acid becomes cholesterol, or lead to a simpler structure from a complex structure. For example, glucose becomes lactic acid in our skeletal muscle. The former case is known as anabolism, and the latter case is known as a catabolic pathway. Anabolic pathway, as expected, consumes energy. This consumes energy. For example, assembly of protein from amino acid, they require energy input. On the other hand, catabolism, a protein from amino acid, they requires energy. The catabolic pathway lead to release of energy. For example, when glucose is degraded to lactic acid in our skeletal muscle, energy is liberated. Okay. Now come to enzymes. What are enzymes? Almost all enzymes are protein. There are some nucleic acid that behave like enzyme. They are called as ribozymes. Okay. One can depict an enzyme by a line diagram. Okay. An enzyme, like any protein, has a primary structure, that is amino acid sequence of the proteins. An enzyme, like any protein, has secondary and tertiary structure. Okay. An active site of an enzyme is a okay, we will discuss it later. So what are the characteristics of enzyme? Protein that catalyze biochemical reaction in living cells. Each enzyme catalyzes the reaction of one substrate. Each enzyme requires a specific pH and temperature, and they are the accelerator of reactions. Okay. Okay. What are the similarities between enzymes and inorganic catalysts? Catalysts, they remain unchanged at the end of the reaction, and they can be used again. Secondly, they are required in far less quantities as compared to the substrate. They do not initiate a reaction but rate of reaction by lowering activation energy. They do not alter equilibrium of a reversible reaction. They form short-lived complexes with substrates. Okay. Now, what are the differences between enzymes and inorganic catalysts? All enzymes are protein and they have complex molecular organization. Whereas inorganic catalysts, they are usually small and simple molecules. An enzyme catalyzes only a specific reaction, whereas inorganic catalysts, they can catalyze a number of reactions, hence they are not specific for any one reaction. Third, enzyme action can be regulated by specific molecules. Inorganic catalysts, they cannot be regulated by any other molecule. Enzyme, they are more sensitive to changes in pH and temperature of medium. Inorganic catalysts, they are very less affected by changes in pH and temperature of medium. Nature of enzyme action. There are the 4 steps that are involved in the enzyme action. The enzyme and the substrate are in the same area. Some situations have more than one substrate molecule, then an enzyme will change. Second, then enzyme grabs onto the substrate at a special area called the active site. One such pocket is the active site. An active site of an enzyme is a crevice or pocket into which the substrate fits. Okay. Thus, enzyme through their active site catalyze reaction at a very high rate. Okay. Enzymes are very specific and they don't just grab onto any molecule. The active site is a specifically shaped area of the enzyme that fits around the substrate. I'm again repeating that they are specifically shaped areas of the enzyme that fits around the substrate. The active site is like the grasping claw of the robot on the assembly line. It can only pick up one or two parts. Third process is called as catalysis happens. Catalysis is when substrate is changed. It could be broken down or combined with another molecule to make something new. It will break or build chemical bonds. When done, you will have the enzyme product complex, that is EP complex. Okay. And ultimately, enzyme releases the product. When the enzyme lets go, it returns to its original shape. It is then ready to work onto another molecule of substrate. Now we will continue to discuss about the progress of reaction. When plot is graph is plotted between energy and the enzyme, this is the activation energy with enzyme and this is the activation energy without the enzyme. As you can see, this is the activation with enzyme. Okay, so that means that enzyme accelerates the reaction. Mechanism of enzyme action, as I have told you, that enzyme, they don't just grab any molecule, they are very specific. Suppose log is an enzyme. Okay, and key is a substrate molecule. As we know that particular key fits onto the particular log. Likewise, enzyme and substrate, they also fit specifically. Now we will read about the nomenclature of enzymes. Adding suffix "ase" to the substrate onto which they act. For example, sucrase. Sucrase means it is acting on the sucrose, and sucrase means ase is defining that it is an enzyme. Protease means it is acting on the protein. This enzyme is acting on the protein. According to physiological activity, it catalyzes, for example, oxidase, dehydrogenases, decarboxylases, etc. According to source from which they are obtained, for example, papain and bromelain. Okay, some have been named like ptyline. In ptyline, P is silent, and trypsin, etc. So always note that thousands of enzymes have been discovered, isolated, and studied till time. Okay, most of these enzymes have been classified into different groups based on the type of the reaction they catalyze. Enzymes are divided into six categories or six classes, each with the 13 subclasses and named according by the four-digit number. We won't be discussing about the 13 subclasses, but definitely we will be discussing about the six classes, six broad categories of it. Okay. So, the first category or the first class of enzyme is oxidoreductase dehydrogenase. Enzyme which catalyze oxidoreduction between two substrate S and S dash. Okay, S and S dash. This is S and this is S dash. Is in the reduced form, whereas S dash is in the oxidized form. After the reaction, it got converted, S reduced got converted into S oxidized, and whereas S dash oxidized got converted into S dash reduced. Okay. Cytochrome oxidase, it oxidizes cytochromes, and glycolate oxidizes glycolate. Now, the second category of enzyme is transferases, enzyme catalyzing a transfer of a group other than hydrogen, sorry, okay, between a pair of substrate S and S dash, okay. Suppose this is Sg in the combination plus S dash, another reactant. After the reaction, product formed is this. That means S got segregated and G bonded pair with S dash. Okay. So it is saying that the enzyme involved is glutamate pyruvate transaminase. This has catalyzed a transfer of a group other than hydrogen between a pair of substrates. The third class of enzyme is hydrolases. Enzyme catalyzing hydrolysis of ester, ether, peptide, glycosidic CC and C halide and PN bond. Okay. It catalyzes breakdown of a larger molecule into smaller molecules with addition of water. Now come to the fourth category of enzyme, that is lysases class. It catalyzes cleavage of a specific covalent bond and removal of a specific group without the use of water. Histidine decarboxylase cleaves histidine into histamine and carbon dioxide. Okay. Suppose this XC is bonded with each other, and this C is bonded with an adjacent carbon atom, which is again bonded with the branched Y atom. After the reaction, the branched atoms they got bonded with each other, and carbon carbon they forms double bond with each other. So in short, you can say that they catalyze removal of group from substrate by mechanism other than hydrolysis that leaves the double bond. Okay. Now come to the fifth category or the fifth class of enzyme, that is isomerases. They include all enzyme catalyzing interconversion of optical, symmetric or positional isomers. They catalyze rearrangement of atom in a molecule to form isomers. For example, phosphohexose isomerase, they convert glucose 6-phosphate into fructose 6-phosphate. Okay. Now the last class of enzyme, that is ligases, enzyme catalyzing the linking together of two compounds. For example, enzyme which catalyze joining of CO bond, CS bond, CN bond, PO etc. bonds. They catalyze covalent bonding between two substrates to form a large molecule, mostly involving utilization of energy by hydrolysis of ATP. Rubisco biphosphate carboxylase, that is RUBP, which is the most abundant protein in biosphere, as I have told you in the previous section of this chapter. So RUBP carboxylase, it catalyzes the joining of RUBP and carbon dioxide in photosynthetic carbon fixation. This is the example of ligases. Now the catalytic cycle. Catalytic cycle are made up of 4 steps, or 4 steps are involved in the catalytic cycle. Substrate, it binds to the active site of the enzyme, because enzyme, they don't just grab any substrate molecules, so they both are very specific to each other, just like lock and key. Second, binding of substrate induces the enzyme to alter its shape and fit more tightly around substrate. Okay. Active site of enzyme now are in close proximity of substrate, that breaks the chemical bond of substrate, and an enzyme product complex is formed, that is EP is formed. Enzyme releases the product of reaction, and free enzyme is ready to take up another molecule of substrate, because they can be reused in any other reaction too. This is the diagrammatic representation of enzyme and substrate. As you can see that this is enzyme, this pink part is enzyme, and this is the substrate is maltose. Okay, and this is the active site of it, this is the active site of it. Okay, active site of an enzyme is a pocket into which the substrate fits. This enzyme, through their active site, catalyze reaction at a very high rate. Okay, so it got just like lock and key, this substrate got bonded to the substrate enzyme, and thus product is removed in the further reaction. The product is removed, maltose is broken down into its monomer unit, that is glucose and glucose. Okay, can you control them, students, can you control them? Yeah, it's a good question. I know what you are thinking. What if enzymes just kept going and converted every molecule in the world? They would never stop, they would become devils, isn't? Don't worry, there are many factors that can regulate enzyme activity, including temperature, activators, pH levels, and inhibitors. Okay, factors that affect enzyme action. First is temperature. Temperature, they show highest activity at optimum temperature. Activity declines above and below the optimum value. pH, they are very specific to pH. Enzyme function in a narrow range of pH. Highest activity at optimum pH is shown or seen. Concentration of substrate, the velocity of enzymatic reaction arises with the increase in substrate concentration till it reaches maximum velocity, that is Vmax. Further increase of substrate does not increase the rate of reaction, as no free enzyme molecules are available to find with additional substrate. Fourth is enzyme inhibition, in which when the binding of a chemical shuts off enzyme activity, the process is called as inhibition, and the chemical is called as inhibitor. Competitive inhibitor, which closely resembles the substrate in its molecular structure and inhibits the enzyme activity. For example, inhibition of succinate dehydrogenase by malonate. The factors that affect enzyme action, that we will discuss it in detail. First, we will be discussing about the temperature. Enzymes generally function in a narrow range of temperature and pH. Each enzyme shows its highest activity at a particular temperature and pH, which are called as optimum temperature and optimum pH. Activity declines both above and below the optimum level. Low temperature preserves the enzyme in a temporarily inactive state, whereas high temperature destroys enzymatic activity because proteins are denatured by heat. Okay. Effect of pH. So what happens, the pH? Each enzyme showed its highest activity at a particular pH, that is known as optimum pH. Activity declines both below and above optimum value. Low um, if we plot a graph between increasing enzyme activity and pH, then we will get a optimum pH, below and above which activity declines. Now the effect of the substrate concentration. With the increase in the substrate concentration, the velocity of the enzymatic reaction rises at first. Okay, as you can see that it is rising. The reaction ultimately reaches the maximum velocity Vmax, which is not exceeded by any further rise in concentration of the substrate. This is because the enzyme molecules are fewer than the substrate molecules, and after saturation of these molecules there are no free enzyme molecules to bind with the additional substrate molecules. Okay. Effect of chemicals. When binding of a chemical reduces or shuts off the enzyme activity, the chemical is called as inhibitor. They are of two types, competitive and non-competitive. When inhibitor closely resembles substrate in molecular structure and it binds to the active site of the molecule, and non-competitive when inhibitor does not compete with the substrate for active site. Okay. Competitive inhibitors are succinate and malonate. This is the normal binding of the substrate, where the substrate binds with the active site of the enzyme and not with the allosteric site. This is the allosteric site and this is the active site. Okay, and these are the action of the enzyme inhibitor, where competitive inhibitor, it occupies the active site of an enzyme, and because of which substrate won't be able to bind with the active site of the enzyme. And this is the altered active site, where non-competitive inhibitor binds with the allosteric site, and due to which in substrate won't be able to combine with the enzyme. Feedback inhibition. Enzyme activity is inhibited by the product of the same enzyme reaction. Glucose 6-phosphate, they inhibit reaction of the hexokinase, and thus it catalyzes phosphorylation of glucose. Okay, this is the start of the pathway, where substrate or reactant got converted into intermediate 1, and then converted into intermediate 2, and then product. Okay, enzyme 1, 2 and 3 are involved. This product will then inhibit this pathway. So you can say that presence of a product that inhibits enzyme 1. What are the cofactors? Cofactors are non-protein constituents found to the enzyme to make it catalytically active. Protein portion of enzyme is called as apoenzyme. Prosthetic group are organic compounds that are tightly bound to the apoenzyme. For example, heme and peroxidase and catalase. Coenzyme, organic compounds which loosely bind with enzyme. For example, NAD and NADP. Metal ions, they are required for enzyme activity. They form coordination bond with side chains at active site and that with the substrate. Okay. So enzymes are of two types, simple and conjugate enzymes. A simple enzyme, they are made up of one or several polypeptide, whereas conjugate enzyme, they has non-protein moiety with polypeptide chain. So it is formed from the combination of non-protein moiety and polypeptide chain. Okay, this is apoenzyme and this is cofactor. This is the non-protein moiety and this is the protein portion. So when it gets bonded with the apoenzyme, it forms a holoenzyme, or that is known as whole enzyme. This is the representation of a holoenzyme. This is the cofactor. This is the catalytic site, and this is the coenzyme that which is bonded with the apoenzyme to form a holoenzyme in all. Cofactor, they have three types: prosthetic group, coenzyme and metal ion, that we have already discussed. Prosthetic group, they are tightly bound to apoenzyme. Coenzyme, they are bound to apoenzyme during course of catalysis. Metal ions form coordination bond with side chains at active site of enzyme and substrate. Example of a prosthetic group is heme. Example of coenzyme is NAD and NADP. And example of metal ion is zinc. So this comes to an end. In the next class, we'll be discussing on animal kingdom. So stay tuned for something very awesome. Thank you.